Hello, today's topic is electrochemical transducers and biosensors. I'm Dr. Solomon Waneri of the Department of Biomedical Engineering, University of Lagos. Our lesson objectives are to explain the functions of electrochemical transducers, describe the main features of the different types of electrochemical transducers, explain the main features of electrochemical biosensors and their applications, describe the working principle of electrochemical biosensors, describe how a diabetes test strip works. Electrochemical transducer. Electrochemical transducers are devices that convert a chemical signal into an electrical signal or vice versa. So basically what it does is that it makes use of an electrolytic solution to convert a chemical signal into an electrical signal. This is quite applicable in healthcare, and one of the healthcare applications is in the use of the diabetes test strip for the monitoring of glucose level in the blood. So it has a wide range of applications. Also, an electrochemical transducer uses a chemical change to measure the input parameter. Usually when an input parameter is sent to an electrochemical transducer, a chemical change occurs. A chemical change occurs. And usually the output is usually in the form of electrical signal. The output is usually in the form of electrical signal or a changing electrical signal, which may be in form of current or voltage. The output of an electrochemical transducer is usually a changing electrical signal, which can be in form of current or voltage. There are basically two types of electrochemical cells. We have the galvanic cell and the electrolytic cell. The galvanic cell and the electrolytic cell. Let's look at the galvanic cell. A galvanic cell is also referred to as a voltaic cell. A galvanic cell is also known as a voltaic cell. A galvanic cell was named after Luigi Galvani and Alessandro Volta for their immense contributions to the understanding of electricity and chemical reactions. It is an electrochemical cell that converts chemical energy stored in spontaneous radius reactions between two substances into electrical energy. So what happens in a galvanic cell simply is that it's a cell that has electrodes and also an electrolytic solution that converts that electrolytic solution into electrical energy. That is basically what happens in a galvanic cell. The main components of a galvanic cell are: 1. The electrodes. A galvanic cell consists of two electrodes, an anode and a cathode. Oxidation usually occurs at the anode. You know that oxidation is the loss of electrons. So at the anode, electrons are lost, while reduction occurs at the cathode. Reduction is gain of electrons. So when electrons are usually gained at the cathode. Then we have the electrolytes. In the electrolytes, we have the electrodes immersed in the electrolytes. The electrodes, are the anode and cathode. The anode is the positive electrode, while the cathode is the negative electrode. Both of them are immersed in an electrolyte solution that contains ions of various substances. The electrolyte allows the flow of ions between the electrodes to maintain charge neutrality. That is basically what happens. The electrolyte allows the flow of ions. Then the third one is a salt bridge. A salt bridge is used to connect the two half cells. It helps to maintain electrical neutrality. That is basically what a salt bridge does. It helps to maintain electrical neutrality in the cell. The next is electrolytic cell. Electrolytic cell is the second type of electrochemical cells. An electrolytic cell is always supplied with electrical energy from an external source. That is the main difference between an electrolytic cell and a galvanic cell. A galvanic cell depends on the electrolytic solution inside and the electrodes that are placed in it, while an electrolytic cell depends on the external source of electrical energy. An electrolytic cell undergoes both oxidation and reduction reactions, usually the redox reactions, when electrical energy is passed into it. An electrolytic cell is obtained by connecting a source of direct current into an electrolyte through electrodes. So the direct current is provided by the batteries, whose positive end is connected to the positive electrode. Yes, what happens in an electrolytic cell is that we have batteries that are external batteries that are connected to the electrodes in the electrolytic cell. The positive terminal of the battery is connected to the anode, the positive electrode inside the electrolytic cell, while the negative terminal of the battery is connected to the cathode in the electrolytic cell. So this is how an electrolytic cell is. We can see that this is the positive terminal of the battery. It is connected to the anode. This is the anode of the electrolytic cell. So the positive terminal, the longer terminal is the positive, and positive is written here. The positive terminal is connected to the anode, while the negative terminal is connected to the cathode. When the switch is on, electrons flow from the negative end of the battery into the cathode. The concentration of electrons on the cathode causes the positive ions in the solution to migrate to the cathode. Yes, because there are so many electrons in the cathode, the negative ions also move to the anode. At the cathode, one of the positive ions pick up electrons and become reduced. Yes, that's what happens at the cathode. We have some positive electrons at the cathode. They pick up the negative electrons that migrated and they become reduced. No, reduction is gain of electrons. So while at the anode, one of the negative ions loses the same number of electrons picked up and becomes oxidized. Oxidation is loss of electrons. So the lost electrons are taken away through the wire back to the batteries. So those lost electrons are taken out of the electrolytic cell and they move back to the batteries. Then when that happens, the bulb lights up, indicating that the circuit is complete, which means that the solution is a conductor of electricity. So an electrolytic cell can generate sufficient voltage to power a bulb. Now let's look at electrochemical biosensors. Electrochemical biosensors are a class of biosensors that function using an electrochemical transducer. They function using an electrochemical transducer. They can detect biological materials like enzymes. Electrochemical biosensors can detect various components just like glucose in our blood. The electrochemical transducers can detect that. They have been used widely in clinical diagnostics, environmental monitoring, and food safety due to their specificity, sensitivity, and ability to provide real-time measurements. Yes, they have very high specificity. When a measurement system has a high specificity, what it simply means is that that measurement system can help us to have very low false negative. It means that the false negative is very low. And while when it has high sensitivity, you can correctly predict those that have a certain disease. For instance, in the case of diabetes, if that biosensor has high sensitivity, it can correctly predict diabetics in those that do the tests using the test strip. So the main components of electrochemical biosensors are: 1. The biological recognition element. The biological recognition element. The biological recognition element is a biomolecule that interacts selectively with the target analyte. Yes, what happens is that a biological recognition element can interact with the analyte. Let's take, for instance, when doing a test, a diabetes test with a glucometer. The analyte in this case is the glucose in the blood. So when blood is extracted from the patient, the biological recognition element makes it possible is able to interact with that glucose that is in the blood. The biological recognition element is able to interact with that glucose that is in the blood. That is the solution of interest. Examples include enzymes, antibodies, aptameters, and whole cells. Then we have transducers. That's the second component in an electrochemical biosensor. A transducer is electrochemical biosensor. In electrochemical biosensor, converts the biological signal into an electrical signal. That is what a transducer does in an electrochemical biosensor. A transducer converts that biological signal that you have extracted from the patient into an electrical signal like current or voltage. Let's look at the working principle of electrochemical biosensors. The working principle of electrochemical biosensors involves the specific binding of the target analyte to the recognition element. Yes, the target analyte is the quantity that we want to measure, the quantity of interest that we are interested in finding the value. That is the target analyte. It's able to interact with a recognition element that is able to identify the target analyte. And usually this recognition element could be an enzyme. And this leads to a measurable change in electrical signal. So when the target analytes interacts with the recognition element, there usually a change in the value of the electrochemical signal. For example, in glucose biosensors, glucose oxidase catalyzes the oxidation of glucose, and the resulting change in current is proportional to the glucose concentration. So, while using a glucometer, I will explain it when we get to the next slide. Yes, so let's look at how a diabetes test strip works. That will help us to explain a basic electrochemical biosensor. A diabetes test strip is often used for monitoring the glucose level in the blood of a diabetic. So it usually involves an electrochemical reaction, and the electrochemical reaction helps us to measure the concentration of glucose in a sample. So when a person wants to do a test to monitor the glucose level of a diabetes patient, usually that's what happens is that it's an electrochemical reaction. That test will help us to identify the amount of glucose in the blood of that patient. The glucose is the analyte. The glucose in the blood of the patient is the analyte. So let's look at the various steps. The first step, the test strips are used in glucometers to measure blood glucose level in diabetics. So usually the test strip is used to measure the glucose level. The first step usually involves pricking the finger with a pin, obtaining a small blood sample. Yes, when you prick the finger of the person, you can obtain a small quantity of blood. Then when the blood sample is applied to the test strip, at the tip of the test strip we have a small electrode. So the glucose in the blood usually reacts with the enzyme on that small electrode on the test strip. That is what happens. So when the blood from the test strip drops on, when the blood from the patient drops on the electrode in that test strip, a small electrode at the tip there is an enzymatic reaction. The enzyme usually catalyzes the oxidation of glucose. The glucose is oxidized, producing gluconic acid and hydrogen peroxide. That is what happens. Once the blood drops on that test strip, on the electrode inside that test strip, an oxidation reaction takes place. And that oxidation reaction produces gluconic acid and hydrogen peroxide. The hydrogen peroxide then reacts at the working electrode. And what happens is that the hydrogen peroxide generates an electrical current. It generates an electrical current. Now, the electrical current will now be proportional to the amount of glucose in the patient, which means that a patient that has a very high glucose reading, what it simply means is that the amount of current that will be generated will be more than that of a patient that has a very low glucose reading. That is very important. So because of the proportionality, as I said earlier on, that the electric current that is generated is proportional to the amount of glucose in the blood. So the higher the amount of glucose in the patient, the higher the amount of current that will be generated. And that is basically how it works. So the value is now displayed on the display of the glucometer. So for a patient, for instance, that has a blood glucose level of about 125, about 125 or thereabout, his level is usually higher than the amount of current in the glucose level is higher than someone that has a lower level. That is simply how it works. So in conclusion, we have examined the functions of electrochemical transducers, and their basic function is to convert chemical signals into electrical signals. And we have also identified the two main types of electrochemical transducers, the galvanic and the electrolytic cells. Also in this class, we have discussed the electrochemical biosensors and their applications. We have described the working principle of diabetes test strips as biosensors. Thank you for listening. These are the references and further readings.